Thank you for this introduction, Delphine. So Maciej is the proper one, but whatever you do is it's fine. Um, so I would like to start by saying that I'm super grateful to be among so many jelly crazy people, and this was an amazing pleasure to listen to all your amazing talks. So the common theme among many of the talks that we've heard so far was that people were looking into exploring jellyfish biology by taking account of the environment. So try to figure out how the environment shapes the jelly biology. We heard a talk on how like winds accumulate jellies or how the temperature promotes outbreaks of or jelly blooms. My idea is quite opposite. So I would like to suggest that we could also use jellyfish biology to try to infer the state of the ecosystems. And for that, I will be using the Arctic Ocean as my model system. So first things first, climate change is real. Temperatures are rising globally, but there's a particular place in the world's ocean that's warming nearly 3 times faster than the global average, and this is the Arctic Ocean. And the reason behind that, it's called Arctic amplification. So this all comes down to the fact that the more warmer water gets to the Arctic, the faster the ice melts, the more dark ocean it uncovers, and the dark ocean can absorb more of the heat from the sun, which warms it up even faster. But where do we get warm water in the Arctic Ocean from? From the Atlantic Ocean. So there's this huge connectivity between the Atlantic Ocean that delivers the warm and more saline Atlantic water with the Gulf Stream or its northward extension directly to the Arctic. So the Gulf Stream then splits into two branches nearly at the top of the continental Norway. One branch would flow through the shallow Barents Sea, where it loses most of its heat to the atmosphere. And then there is this very important branch that goes through the deep Fram Strait, and it fuels basically the entire circulation of the Arctic Ocean. So what we've seen in recent years is that this water flow of the Atlantic waters, it's becoming more warmer, more saline, and then there's this bigger volume of Atlantic waters getting delivered higher into the Arctic. And this obviously comes with the cascade of environmental effects. So what we're seeing now is that the typically polar environments are now turning into more a boreal state in a process referred to as Atlantification. And Atlantification comes with the magnitude of ecological effects. And the most obvious are biogeographic changes. So we are seeing more boreal species appearing upwards, like in the higher Arctic. But we are also seeing some differences in the reproductive patterns. So boreal species are now able to reproduce in the Arctic, but they are also even able to reproduce 2 times in the Arctic, much like in temperate zones. But as is usually the case, we know all of that based on the studies on crustaceans, and the literature on gelatinous zooplankton is very scarce, if any. So to tackle my big question, whether jellies can be used to observe or monitor the Atlantification of the Arctic, we first needed to come up with the diversity baseline of jellyfish in the Arctic, and then study their population structure as a potential another indicator of the Atlantification, and then try to link that with environmental drivers that we could sample in the Arctic. So since going to the Arctic is rather expensive, we decided to use samples that were already collected as a part of the Polish monitoring of Arctic zooplankton. So I picked up a 12 years long time series of plankton samples collected from the area spanning nearly 300,000 square kilometers in the European Arctic. So I got the plankton samples and the CTD probe readings from this area. And then I've also added some samples collected by a friend in 2012. They were collected in the vicinity of my research area, but with the multi-net plankton sampler. So he went deeper down to 1 kilometer water deep, so to get like a better baseline of the diversity in the region. And I must note here that all those samples were previously analyzed, the papers were published, but the taxonomic resolution in jellyfish was basically jellyfish. So they counted jellyfish without going deeper into what species were present there. So this was basically useless for us, but this is a common case for regular plankton monitoring, that people don't care about jellies. So my task was basically picking all the jellies from the samples, identifying them. So we got nearly 15,000 records of jellies from the region. and then measuring each and every individual, and then identifying life cycle stages if possible, to get the proxy of the population structure of different species in the area. Since we are limited on time, I will not go into much detail, but if you want to learn more, I invite you to read some of the papers that resulted from this work. And so just to give you I try to be brief overview of what we found. So to start with the diversity baseline, we identified 26 taxa, and they were all like species that we expected to see there. So there was nothing new, nothing surprising there. We see highest diversity in shallow shelf waters, primarily due to the presence of meroplanktonic taxa. And then in the open ocean, what we see was high diversity, mainly driven by Trachymedusae. Then, given this diversity baseline, we went to look at the environmental drivers of those distribution patterns. So because of the giant spatial and vertical scope of our sampling, majority of the differences were driven by geographic factors, with the hydrological factors, the ones that we are really interested in, driving only 1% of the variance on their own. But what's very important is that the interaction of the hydrological factors and of other factors explained most of the variance in our data, which was something that we were hoping for. And this just to note that biotic factors, which were basically food abundance for different jellies, were also able to explain significant part of variance in our data. But this was not very important. What's really important is the fact that we found some species that were strongly associated with particular conditions, like this siphonophore, Physophora hydrostatica. We found it only in super shallow and super warm waters in the Arctic. And then what's even more interesting is that some species, like some developmental stages of the same species, showed proclivity to different environments. So they had different environmental preferences alongside their life cycle. And this is Aglantha digitale. So with that in mind, we went on to apply indicatory species framework proposed by Louis Legendre to test the strength of association of different species towards particular water mass, oceanic fronts, or water mass or oceanic currents. And these are a few examples. So Siphonophore rugiacovia plicata was found indicatory to Arctic water mass, juveniles of Aglantha digitale for the Atlantic water mass, and this nice Hydromedusae Bougainvillea superciliaris was indicatory for polar surface shelf waters. So the next big question was whether our indicator species could mix in between adjoining water masses. And so when two water masses meet, they usually have different temperature or salinity signatures. The oceanic front would form in between. And when the two water masses have different density, then we have something called active oceanic front, which basically means that we have strong density gradient that's impermeable for jellyfish. And we know that from literature that jellies tend to aggregate near the density discontinuities. But what we have in the Arctic is that because we have warm and more saline waters meeting cold and less saline waters, there is basically no density gradients in between them. So we are seeing what's called in literature passive oceanic fronts, not associated with density gradients. And there was no information whether this is permeable for plankton or not. So we start with the regular indicatory species analysis, pointing out to Beroe being typical for the site west of Arctic front, Aglantha for the Atlantic inflow, and then Mertensia ovum being found mostly for the zone east of polar front. And then we took advantage of the fact that heat content of the Atlantic inflow showed the interannual variability. So in extremely warm years, like in 2006, the amount of the heat delivered to the Arctic pushed aside the oceanic fronts. So this allowed us to pick particular sampling sites that jumped from one side of the oceanic front to another site while remaining at the same geographic position. So we used this case to try to see whether we would have the same jelly composition, with the hypothesis here being that if the fronts would be completely permeable, we would see the same composition irrespectively of whether jelly sampling site would be on the east or west side of either of the front. Fortunately for us, that was not the case. So we found that even those not density-driven oceanic fronts, they can structure jellyfish community. So what's very interesting and maybe slightly counterintuitive, the warmer Atlantic waters had a higher proportion of Aglanta digitale, had lower diversity of jellyfish that was basically dominated by Aglanta, and they had also higher abundance. And this could mean something for the future forecast of how the Arctic would look like, but I'll get back to that later. So the final big question that we had was, since Aglanta was emerging as an important indicator of those warm Atlantic waters, we wanted to look deeper into their population structure to see whether the population structure in itself could be an indicator of the progression of Atlantification of the Arctic. So what you see here are the 1 millimeter size classes of Aglanta that we use as a proxy of the population structure. So if we focus on extremely warm years, what you can see is that they were characterized by higher abundance of Aglanta, irrespectively of sizes. And these are the white dots that you can see. But what's very important is that in those extremely warm years, what we were seeing in the southernmost area of our sampling or investigative area was a high proportion of baby jellies. And this may seem counterintuitive. Warmer water should promote earlier onset of the reproductive cycle, so we were expecting to see tons of large jellies. But what this actually means is that our jellies are now reproducing 2 times in this area, so twice a year, which so far was only reported in the north of England. So we are like jumping with the boreal state really quickly to the Arctic. So what also came out of this data was that there is this clear linkage between the situation or the population structure in the lower latitudes versus higher latitudes, clearly pointing towards the connectivity of the populations. So the southern population was seeding the northern population. And we don't have any molecular data. These are ancient samples kept in formalin, but I know that Charlotte Havermans is currently working on the population genetic study of Aglanta, and she's been finding no structuring across the Arctic Ocean. So basically all gets derived from the Atlantic up north. And then the final confirmation, so this was a slight exercise that we did. So we were trying to use the presence and absence data as well as abundances of Aglanta. And we built this simple model trying to delineate the differences in response or ecological response of baby jellies and large jellies. So that's why we introduced this interaction term with all the variables tested. And what this allowed us to do was to delineate how baby jellies and large jellies differentially respond to different environmental patterns. And what we saw here was that we see this very pronounced response from the baby jellies to changing spatial patterns that we are sampling or changing environmental factors, whereas larger jellies were not as responsive as the babies. So I know that I touched on many topics, so I just, as a quick conclusion of the talk, what I show is that jellyfish diversity, but also their population structure, can be used as a good indicator of Arctic Ocean Atlantification, and most likely for any other oceanic processes that we are observing. And that the passive fronts, that at least those that we see in European Arctic, can form barriers for jellyfish mixing. And then Atlantification will likely promote less diverse and more abundant Aglanta-dominated jelly community in the Arctic. So it seems that jellies are indeed coming to the Arctic en masse. So with that, I would like to thank you all for your attention.